There you go. So I'm going to stop sharing my screen again. And can you see my presentation? Yes, great. So today we're going to talk about the ethical aspects of modifying an image. We're going to talk also about how to make a figure and how to make a good figure, a scientific figure for publication, and also how to write the materials and methods. This session is going to be a bit shorter than the previous one, and it's going to be more straightforward. There is some exercises that I will do on my own computer because it requires specific softwares, and so I will do, and you will be able to follow what I'm doing on the screen. So first thing is about the ethical aspect of microscopy. What can you do and what are you not allowed to do? There is several things. There is things that you have to do. You must follow some of the rules, and you must make sure they are all done properly. There is things you are authorized to do, and there is things that are prohibited. So I'm going to just review quickly these three of them, and then give you some resources to get more information about what can be done and what can be done. First, what the things that you have to do. This is valid in science, and it's valid for microscopy, but also in science in general. It's as soon as you're publishing something, you are responsible for the data you are publishing. So you are responsible for the images, the analysis, and the conclusion that you are drawing from this data. If you get wrong, but then it can only be your fault. There is no excuses, or you cannot say, "Oh, I didn't know." When you are publishing something, you must know, and you must find the information to know it. So there is a part of responsibility, and that's why people like me and others are here, is just to ensure that when you are publishing, you are doing things in the proper way, and so people cannot go back to you and say, "Oh, you didn't do right." So that's the part of the responsibility that you must know when you are publishing. The obligations are to keep the original data. So again, here there is no excuses. It was back in the days when we didn't have a computer storage, but now we have a lot of computer storage, and we are working with digital data only. So you must keep the original pictures. And when I talk about originals, I talked about the one that comes from the microscope or the instrument, whatever you are using, and you have to keep them in the original state without any modification, with the metadata from the instrument, but also from the experiment. So if you call your image one, you must have somewhere a file or in a digital logbook where you say image one correspond to my control animal, third image, or whatever. So we need to be able to kind of identify what are these data. So that's what we call metadata. The second thing is we need to be able to trace things down. So if you publish a picture and you quantify it, we need to be able to follow which image is giving rise to which data. And it works in both ways. If I show you a point on your graph, you should be able to find the images that gave rise to this point. And this is what I call traceability. So you need to be able to do both ways, to get from the picture to the data and from the data to the picture. And the last thing you have to do is to make the data available. There is online databases where you can store your data. It's not free, but it's highly recommended to do that. Omero is one of example, but it provides a framework where you can actually send your data and they will store it, and you can publish, and people can consult the data that you have been publishing in the raw format. You also have to make sure that you make them available when people ask you. So I want to review your data. I want to do another analysis on the data you generated. If I sent you an email, then you should be able to send me a link with the exact same pictures that you have been taken. This happened more and more, and actually there is some registry online where you can actually pull the data and you can actually have access to them to reanalyze them. It's more into the field of cryo electron microscopy, not so much in the field of fluorescent microscopy, but I believe that it's going to happen more and more. Then I'm going to just review things you can do and things you cannot do when you are playing with your images. We said that we do a lot of image processing, image extraction, data analysis, and so on. So various things you can do. For example, you can adjust the brightness and the contrast. That's okay to adjust the brightness and the contrast, especially when you know that on 16 bits images, you cannot see most of a signal. So it's not that you can, it's you have to. Otherwise, the image is going to be mostly black for fluorescence images. You can also use non-linear adjustments such as the gamma. There is some things that apply to that, but it's generally okay to do that. And things that I put in authorized, but it's actually a requirement. You have to, it's an obligation. You must treat or process all the images from both conditions the same way. If you are comparing your control and your mutant, and you say, oh, my mutant was, the signal was stronger, so I decrease the brightness because I want to look the same. This is something you cannot do, and so you have to process the images the same way. And then cropping is generally okay. You can crop an image to focus on the area of interest. So this is usually okay. Things you cannot do, it's, for example, you cannot modify a part of an image. So if you are looking at, I don't know, of a tissue, for example, a brain slice, and you say, oh, this region it's too bright, and I have to bring it down because I cannot see my signal otherwise. So you cannot select this area and say, okay, I'm going to adjust the brightness just on this region. This is not okay. You cannot also compare images if they have different display levels. So this is exactly the same than not treating your images the same way. So if you want to compare them, you have to use the same display options. And then it's not okay to crop images. So then you're going to say, oh, you tell me it's okay, and then you tell me it's not okay. So what's the deal here? Well, the deal is that first, all the things you are done are conditional to two things. And the second, well, I'm going to say the two things, and then I'm going to explain to you why cropping is not okay. The first condition is that any modification, processing, treatment you do to your images must be reported in the materials and methods. This is not the case most of the time, and it should be done. So don't get yourself in troubles, and just put everything you have done, and people will be able to follow up the processing of your images. The second thing is you cannot do a modification that's going to change how you perceive or interpret the image. If it changes the conclusion you want to draw from the original image to the final image, then it means you cannot do that. An example of cropping is like I'm comparing a control situation where I have cells on my image, and I'm going to crop on one single cell. I have the same thing in my tests condition, and I say, okay, all the cells around are very bright, but I want to show that the signal decrease. So I'm going to crop on the cell that is has a lower signal. This you cannot do. If it changed the conclusion, then you cannot do that. So cropping, for example, it's really, and actually all the things you do are conditionals to these two things: reporting and not changing the interpretation of the data. I put some resources here, but most of them come from this guy, Douglas Cromie from the University of Arizona. He published something called Avoiding Twist Pixels, an ethical guidelines for data usage and manipulation. And I put the link here. He wrote quite some interesting things, and he also wrote this kind of what's in a picture, temptation and image manipulation. And in this article, what it shows is that there is sometimes data that's published like that, some cells, and you can actually take this data and process them to display if there is any kind of trick that has been done. So in this case, the person who was publishing it selected the cells here, and because they were, I guess, of lower brightness, change the brightness to display a bit more even image. Yeah, there is a question. I see someone in the chat writing, but I cannot read it. So Douglas also put an online learning tool, and I put the link here. You will have the access to the PowerPoint from the last lectures. I will send it to you so you will be able to consult these resources. So now we're going to move to the second section, which is composing figures for scientific research. And I made a poll. I don't know how to put the poll out. Yeah, I'm going to...
Nicolas?
Oui.
Oui, c'était pour la question dans le chat, tu disais que tu ne pouvais pas la voir?
Oui.
OK, dans le fond, la personne dit gamma is included in the modification that should be reported.
Okay, so Monique was saying that the gamma it should be included. If you modify it, yeah, then you have to report it. So gamma is a nonlinear adjustment that can change the display of an image, and since it's nonlinear, it has to be reported because it's very hard to kind of explain it if you don't give the number. I made a small poll online, so you can actually see the result in live because I'm sharing my screen, and basically it's how many people here have published pictures and figures, scientific figures, and how many of them, if so. So you can see the numbers. So I guess we have some people quite experienced with figures, and others that are beginner yet. Okay, so that's the end of the poll. The second, making a figure, there is actually 2 steps to make a figure. The first thing is you have to prepare the images you want to include in your figure. And I'm going to give a brief overview of how to select them and how to prepare them for that. And the second part is actually to build up the figure. And I'm going to give some example and show you how to do that in a proper way. So the general procedure to prepare your image, it's I will show it in a second, but this is the most delicate part because this is where manipulation can be done in a wrong way. So you have to be very careful before you prepare, while you are preparing your image, not to do something that's gonna decrease the amount of data or change the data or change the conclusion that you want to show. The first thing you need to do is you have to use the right software. And I highly suggest people not to use Photoshop because Photoshop is not meant to modify scientific images. And in Photoshop, you have a lot of tools that are meant for modifying an image to look good. And this is not what we want to do in science. We want to keep the data and look at real things. So I highly suggest to use ImageJ and Fiji, which have a lower numbers of modifying pictures, and they are meant for scientific images. And so I highly suggest you guys to use that to do the modification you need, and not use Photoshop because the risk is too high to use a wrong tool, such as clone and stamp. Oh, I have this bubble here, I want to make it disappear. This is not okay. And in Photoshop, it's very easy to do, but this is not okay. A bubble is okay. You can leave bubbles. An imperfect image is perfect. So yeah, the first thing after when you decided to use your software, then you need to select the images. And I'm talking about the original
screenshot de l'écran.
Pardon?
Aménagé.
Geneviève? Ah. Bon, pas grave. Je continue. So the first thing is to select the images. I'm talking about the original images that are representative of your data. Since you have done a lot of acquisition and you do a quantification on this image, it's quite easy to actually pinpoint the one that are around the mean. So if you are working properly, then your data is traceable. So you can actually say, okay, this value here on my graph correspond to these images. I'm going to go and find this image. So if you're working properly, then everything should be smooth. And the best thing to do is actually, if you are putting a graph, I suggest you to put a plot dot plot. So you have all the data appearing. You just don't have just the mean. And you can actually color. Oops. You can actually color the. I'm just gonna stop that so I can stop the quiz. And then share the result. There you go. And I'm going to come back. There you go. There you go. Okay, can you still see my screen? Good, good, good. So to select the images, and if you did all the analysis in a proper way, it's quite easy. So what I suggest is actually to use a different color for showing the image you are showing. So if you have a graph with a lot of dots, just take the dot that belongs to the image you are showing and put it in another color. So showing, okay, I'm showing you this particular data point. This makes things really trustable, and people can move from the example and to the graph. It gives a image view of what a graph would present. The second thing is to calibrate the intensity of an image. This is it's true when you are just showing one example of an image, but if you are comparing conditions, which is often the case in research, you just want to make sure that the test condition and the control condition are displayed in the same way. And this is using the calibration features in ImageJ, for example. I will show you in more details in the next slide how to do that. Then you need to select the colors you want to display. In fluorescence, most of the cameras are using grayscales, so you can of course display your image in a grayscale into your figure. You can also use a color you picked. You can use pseudo colors where you actually code the intensity of an image using color gradient, and this is very, very useful because, as you remember, our eyes are not really good into identifying gray levels. We can only see around 50 levels of gray. So using pseudo colors to color the intensity it's a good tool. So we can actually have a better perception of the intensity in the image. If you are using two colors, I highly suggest that you use the combination magenta and cyan, because people are colorblind. Around sometimes 10% of the population is colorblind. So this is really the basic color you should use. If you are using a free channel, then RGB are the most, so red, green, and blue are the one mostly used. But you will have to split the color because it's kind of hard to see overlap or different individual images when you are combining all the colors together. And then when you are done all of that, then you're going to create a RGB copy of this image and save it as a TIFF. So I'm going to give you the layout of the exercise I'm going to run while sharing my screen, and we're going to prepare some images for the acquisition for the figure. So the exercise is to open an image. I'm just going to go quickly to open an image, to adjust the level in the gray levels. Actually to display the gray levels, to adjust the brightness and the contrast. And I will show you how to adjust it manually. And then to proceed the same way to the other channels. And then the most important thing is going to be to apply the same parameters to all the other image that belongs to this data set. So I'm going to stop the presentation now, and I'm going to move to sharing my screen. So I'm going to take these images that has been taken on a microscope that is just on the same floor, just a bit further. It's a simple microscope. It's nothing motorized, but it has good objectives, and if you have good sales, then it's going to be easy. So I have to open them with Fiji, and to open it, I just drag and drop. this launch the bioformat plugin. Bioformat is actually a plugin that opens all images that are not TIFF, like the one from the companies. This one has been made by an icon image. So it's a ND2 format. And using bioformat, you can open them. You can see that in this window, you have the auto scale option that you can actually click or not. I'm just going to leave it by default. You can also choose how to display the different channels. I'm just going to leave the default for now. So that's my image. I don't see much. There is 3 channels, but they are all looking red for some unknown reason. So the first thing I'm going to try to do is to actually display them into a gray levels. And to do that, I go to Image, Color, and I take my channel tool. In real life, I will never go to Image, Color, and Channel tool. I will just use the shortcut, this one, and it will just pop up the window like that. In this window, you can choose how you're going to display the different colors. If you look at Composite Sum, it's going to just put all color in RGB and display the sum in your screen. In my case, for now, I just want to display in the grayscale. And when you display in the grayscale, you can actually select each channel individually, and you can see them in the grayscale. The second thing I want to do is to adjust the brightness and contrast for this image. You see that I have a very bright spot here. I might not be very interesting in this spot, so I might actually crop the image just to remove this bad spot. Is it changing the data? In this case, no, because it's only qualitative. I just want to show you an example. But if I have to compare a mutant and a control, or a test and a control, then I will actually ask myself, is this actually a result, or is this just an artifact? Before cropping, I'm going to adjust the brightness and contrast. To do that, I will use Image, Adjust Brightness and Contrast. In the real life, I just use this command shortcut. And then you have this histogram from, if you follow up the previous classes, you should be able to read this histogram. Here you have the 0 gray level. Here you have the 255 gray level. And the curve that is right there is actually the number of pixels per gray level. So you see that all my pixels are mostly on the left. And the image is mostly dark. This is the display option. Here it's showing you these pixels that are located here. They will be shown with the brightest value, so they will be white. The pixels that are here, they will be the darkest. So I can actually adjust this, and all the pixels that will be below that, they will be completely black. In the other way, if I adjust the maximum, all the pixels that are above that, they will be completely white. So because my image is dark, what I'm going to do is I'm going to play with the maximum, and I'm going to increase it until I pass the limit. So I can see my signal is here. So what I want is maybe something like that, that is not too much or too strong. So here you see that, oops. Here you see that maybe this region is going to be a bit saturated, and I want to avoid saturation in the image. So I'm going to maybe go something like that. So just be at the limit of saturation for the pixel of interest. The second thing I really like, and I encourage you to do, is to adjust the value, the default value for the black. Most of the cameras, actually, they don't use a 0 value. They manually increase the 0 to something above 0, so you can actually adjust it. This particular camera doesn't do that, so you see that most of the pixels are at the 0 value. So we need to correct for that. And the way of correcting it is to actually adjust the brightness. If you move the brightness, you see it's going to slide the value that I set up here towards the left or toward the right. So what you can do is actually slightly go into the negative value, so that your black is going to be more white. And if you do that, people will actually have an understanding of what is the display, what is the background, but your background doesn't contain any pixel of values. So if you don't display it, if you put it at all dark, they have no idea if you have a background below that, if you have cells below that. So it doesn't have any contrast points. So my view is to always keep some of the value displayed. So here -2, -3. Most of the newer camera, they don't have this problem because the values are above 0 already. So I did the adjustment for my first channel. I'm going to do exactly the same thing for my second and third channel. So I'm going to adjust the brightest point and move down until I reach saturation. In this case, I'm interested in this little puncta. So I don't want them to be saturated, but sometimes you have no choice. If you want to show the rest of the cell, you have to saturate this. So I'm going to maybe decrease something like that. And there, I think I have something decent. Again, I'm going to modify slightly the brightness so I can show the background, something like that. And I'm going to move to the third channel and do the same thing. And done. And now what I want to do is to actually show what the image is going to look like when you're doing a composite. So when you're doing the composite, you see that the three colors are merged together. In this case, maybe you want to apply a different color to your red. Maybe your red is more important than your green, or the blue is more important. So you can always apply a lookup table. And I show you that before. If you go to image, lookup table, and then you can choose the color you want. So here I'm in the green. Maybe I'm going to say, okay, I'm going to use the cyan for the green. So now you can see that you actually see better with the cyan because there is more light, more pixel, more light coming to your eye. So you can actually choose to change these colors if you want to. When you are done with adjusting these display options and you say, oh, I'm happy with that, you're going to save it. Because the display options are saved within the file, you can actually save them as a TIFF. So I'm going to just say adjusted brightness. And then, as I told you, I want to crop my image. So I think it's time to crop the image. I always kept the raw data. I didn't lose any data in this case. And I'm going to say, I want to just avoid these little dead cells. So maybe I'm going to crop in this region. So I select my square tool and I go like that. Usually it's easier to kind of make a square, so to have exactly the same dimension. It's going to make your figure simple as well. So to make a square, you can actually lock the ratio by pressing the shift key at the same time you are making your square. So now you see that I cannot make anything else than a square, whereas here I can make a rectangle. So you press shift at the same time. And you're going to move it so you select the cells of interest. There. And then you crop it. And to crop it, you go to image crop. And you can save it again. Save as TIFF. Crop. Good. We have our image, but this image cannot be imported directly into your figure maker because the three channels are separated. And this image, you can read different channels, but most of the software we are using, they can read only RGB images. So we're going to create a copy of this RGB image by using image type RGB color. And now you can see that I lost my channels. So I cannot separate my channels. Also, if I adjust the brightness and contrast on this image, it will adjust it for the three colors at the same time. So I cannot adjust, individual colors now. So I'm kind of stuck with that. And I really don't want to adjust a brightness and contrast anymore. I just wanted to crop the image. So I'm going to do File, Save. And you see that ImageJ created a new images and he put this RGB tag at the end. So I'm just going to leave it like that, RGB. Okay. So now the most important thing is to apply the same settings to your images. So let's imagine I have again the same image. So I can open it again in Fiji. So I have again the same image. And I want to make sure that the way I display the red in this image is going to be the same in this image. So I want basically to propagate my display channels or my display settings to all the channels here. And you can do it manually by looking at the values and going back to your image and adjusting exactly the same way. But to make sure that things work properly, I think the best is to use a macro. And there is a small macro code that I put in the presentation, but I'm going to use that now. So you're going to take this text. Well, we can actually read this if you want, but just to save some time, I think it's better. So I don't explain, and you can just go and basically use it. So you're going to paste the macro here. So that's your macro code, and this macro code is basically taking the in front image. It's measuring the brightness and the contrast for the channel that you are selected, and it's going to propagate to the other channel. And it's going to prompt, do you want to do it or not? So I'm just going to run this. It says the display range of the image and it gives you the name of the image is, do you want to propagate it to the other image? In this case, you see that it took the wrong image. My original image is not this one, so it's this one. So now we took the right image. You say, okay, the display range is this value. Do you want to apply it to the other open images? Yes. And now the red in this image corresponds to the red in this image. So now I can do the same thing for the green. Yes. Oops, took the wrong one. Well, I guess you have to reselect this picture every time you do. And then I'm going to do the blue. Yes. Done. So now my channels are adjusted. The only thing that I did bad is to actually, I use the green here and I copy into this green here, but it's not the end of the world. So that's how the macro works. If I had 10 or 100 images open, they will do the same. They will apply it to all of them. Then you need to save this image and it's color adjusted. So I'm going to save as a TIFF and I'm going to put adjusted too. And this is my second image. Done. Any question? I guess not. Then we can go on and continue with the next. Yes, it's independent of the deep bit depth, but the macro I used is channel by channel. I wrote a more complicated macro, but I didn't show, where it's measuring the number of channels you have and applying the same parameters to all the channels, so you don't have to do it 3 times. But just for the presentation, it was not useful to show that. I just wanted to show that you must apply the same display range to all your images. So that's the macro. And next, we're going to move to the next part, which is making the figure itself. So we have done with preparing the images. So the first question is, what is a good figure, and also what is a bad figure? A good figure in research is one figure, one result, but you can summarize in one phrase or one statement. If your figure is cannot be summarized in one statement, it's usually that you have too many things in your figure, and you should take down or split it into several figures. Back in the days, the number of figures were limited, and some journals always limit the number of figures. I think it's a mistake, and we should not, limit the way we present the result. I think we should have as many figures as the paper or the science requires it. The figure has to be nice, and when you see it, you want to actually dive in and actually try to understand what's going on. It has to follow the left to right reading and the up to down reading organization. If you don't follow that, people are going to be confused and look for the information. I suggest that it should be readable or understandable without having to read the legend. I know that it's sometimes hard because it's a very detailed, but if you do your synthetic work, like if you summarize your work in a nice way, you can be able to make a figure that people can understand with a limited amount of knowledge and without reading the legend. You should use colors, but not too much and not the wrong ones. it should be complete, but not too heavy to read. For the police, the font, you should use Arial or Calibri. It's called a sans serif font because it doesn't have the serif. The serif is the little, you see the little leg here of the Times New Roman. These little legs are very good for writing a manuscript or text in a book, but they are not very easily readable on a presentation or on a figure. Also, you should not use Comic Sans because it was made for kids and it doesn't look professional at all. So you should use Calibri because these are very popular fonts that are present anywhere, so you would not miss the font. If you use specific fonts, such as things that are a bit exotic, you may actually find places where it won't be displayed properly. So be careful with that. You have to write the font in a size that it's readable. If you display your figure at 100%, then you can actually decide what is readable and what is not. It has to be balanced. And yeah, I highly suggest you to learn how to make a cartoon and use a summary cartoon, because the people that are reviewing your figure or the people that are going to be reading your figure or your article, they are usually busy. They have short time to spend. So if you can actually summarize things, you're going to save them time, so they don't have to go through all the detail of the figure. How to make a figure? Well, the first thing is you have to use something called vectorial software. So images are using pixels, and pixels are called rasterized images. So each space is a pixel, and this is encoded with data. In vectorial drawing, it's different. You don't talk about pixel anymore, but you work with a kind of mathematical formula that's going to display something. So it doesn't matter the way you zoom in. The calculation is going to be done according to the zoom you are in, and it's going to make a nice and smooth line. It doesn't matter how much you zoom in. There is some paid software. So Illustrator is the one I use, but there is also CorelDRAW and Affinity Designer. There is also very good free softwares, and GIMP is one of them. Inkscape is good also. I have very few knowledge about using them aside of trying and testing them, but I was using Illustrator because I learned on that initially. So if you have to choose one, I would because you have never made a figure, I suggest use GIMP because it's not too hard and it's very well supported. Then you're going to create a figure. When you do that, you have to choose a color mode. Typically, the images that's going to be displayed on the screen have to be RGB. And the one that's going to be printed, we're going to be CMYK for cyan, magenta, yellow, and black. This is what the printers are using. But today, I don't see anyone printing articles anymore. So even if the journal asks you to send it in the CMYK mode, don't listen to them. Just send them into the RGB, and there is conversion palette that can be done and can be used. Also, you have to define how big is going to be your figure. I usually recommend people to take the full page, and then you're going to decide if you want to do it on half a page or a quarter of a page or one column, depending on the journal you are submitting. But just start with a full plain page, and then you can decide how to arrange it. And all the resolution has to be at least 300 ppi, which is pixel per inch. The pictures you're going to insert, they're going to be much higher than 300 ppi. But then you will have to kind of, so you insert them as a original resolution, but then when you export the figure, you can actually decide which resolution you want. And I'm going to show you an example of that. So when you insert the image, with the RGB image we have created within the vectorial file. And you want to embed the file within the file. So you don't have to create links between files. Then you will have to adjust the images between them and maybe resize them. When you resize them, you don't change the resolution of the image. You just change the size of the full resolution image within the vectorial file. And finally, I'm going to show you how to add vectorial elements such as arrows or drawings on the top of your image. One of the good example I want to show you was published 10 years ago by Deerduff. These guys were looking at specific ion channels called SK channels on specific lumbar motor neurons, which I was studying at the time. And the pictures are great. They are making very nice and easily readable figures. You can see the figure is there. You see the spinal cord. So anyone who has worked in the spinal cord, they know directly where to look at. They made this little overlay to cut the shape of the spinal cord, and they decided to show also the subregion, which are clearly visible here. And then they made an inset here where they show this region, the lamina 10 region. Within this image, they put some arrows to show, okay, this little cell here and maybe this little cell here is of interest. You can clearly see that, you know, the staining is very strong into the motor neurons, but you can also see clearly the staining into the SK channels. Then they focus on the small cell here, and they go even with higher details, and they magnify these places. So they look at this cell and they show, okay, here you have this type of contact in this region, and they are low intensity contact. And here you have this type of contact, and they show you the details. There is scale bars everywhere. And you can see that because the figure was published online into a rasterized image, you can see that it gets blurry as soon as you increase the image. So for the PowerPoint presentation, I kind of enlarge the image, and you can see that it is not clear, it gets blurry because the image is rasterized. Just to show you a bit of nice images. In these images, they show you another neurons, and they show you the puncta, and they make the colocalization between the three of them. I'm just a bit confused because here they say that they're going to show you the KV 2.1 in cyan, but I don't see any cyan. So it's kind of, maybe they were talking about the blue that you're using, but this looks like something different. So it's a bit confusing about the colors, but the pictures are nice and it's well made, and there is the scale bars and everything. So everything is there for the information. I just want to show you that not all examples are good like that. This is another example of a figure I found, and this is not it's not good at all, actually. First, it's very heavy. When I see that, I don't want to read that. I can still see the spinal cord. There is some kind of drawings on the top, but just too much information, and we don't know what information brings to what data. So it's a bit heavy. The colors also are very, you see the green is very strong, the red is very strong. So the choice of colors is not good. There's too much contrast in this image, so things can get very, you don't feel like reading it. There's too much information, and also the spacing is different. You see that all the image has been spaced evenly, but actually this is one group, it's ABC, and then it goes here, DEF. But you know, someone will have maybe read this in all the wrong way. So the reading is not properly done. So this is an example of a bad figure. Here it's another example of a very bad figure showing graphs. Graphs can be vectorial, so it doesn't matter how you zoom inside, you will always have a line. This is an example of a bad figure because all is in black and white. There's too much information. The scale is not good. Here I can barely read what is what. So yeah, this is the size of the character is not good, and it's very hard to read things that are you know, on the side. So you have to paint your head. So this is another example of a bad figure. And this is within the same article. This is a really bad also example where the person shows the test situation, and they put the inset on the top of the current image. So you actually hiding the image with another image. So this is really a bad example. On the supplemental data of this article that I know very well, you can see that there is an issue with the coloring. You see that now the green and the red, they are kind of different color. The blue also is kind of washed. If you compare to the other figures where the blue is very blue like that, the green is very green. This is the CMYK, RGB to CMYK conversion. It happens that this particular journal, they use a different method of encoding coders for the supplemental data. In this case, they converted to CMYK, only the supplemental data, not the main data. So it just changed the color in a bad way. This is another example that I found, but we were comparing pathology images. It's better. You see that it's readable. A, B, C, D, E, F. There is more gap here, but the arrows are just way too small. I can barely see them. The square also are not very good. And the arrow, you see, it's right there, and here I can barely see it. So the arrow is the arrow size is wrong. Also here I can barely see the M and the N because it's too dark at the back. So maybe one should have used the white to display these two letters. Also in this article, no quantification at all, no traceability, no information about what are we looking at in the materials and methods. So that was a very weak figure. This is an example of making cartoons. Making cartoons is not that hard, and it makes things understandable quite easily. This is an article showing, again, motor neurons going into the limb, and you can make these nice cartoons, and you can express or summarize something quite easily. It's not perfect. You can see that there is still things that are not good, such as writing on the side. Writing sideways is not good usually, and here there is no point. You have enough room to write it on the flat. Also, using a different font makes things very weird. So this is not something I recommend. And using gradients makes things look nicer. You see that this little bulb here has a gradient. Here they have a gradient. So this is actually a good thing to use gradients. It makes things look more professional, but you have to use it in a good way. You can see here, but the perspective is different and the gradient is just wrong. So you have to be careful, because as soon as you make mistakes like that, then it can change how the person, how they feel about the data. This is an example that I have made. I was summarizing an article, and I tried to summarize the full article within a single figure using coders. So that's not a too bad example, because the article was quite complex, and it's divided in two, and you can actually try to read the steps and try to understand what's going on. It's straightforward, and it was a good work. But if you look at the article, the original article was very complex. So I think that was not too bad of a work to actually generate this figure from these graphs. Kind of get crazy here, where it turns around and round and round. So yeah, very complex article. This is a better example, and for the ones who spotted, all the example I was showing, except from the other, were from my articles. So I was making maybe the comments of my own art and figure making. This is one of the last I have published. You can see that the information is quite clear. The colors are good. You use gradients, but not too much. Things are simple. You have all the data you need. So this is actually a good example of making a cartoon. And this is another example of article I have published where I was trying to make the figure look nice. I use this coding for the mutant and the control, or the control and the mutant, with blue and magenta, and everything is coded according to. So you can actually follow. It's quite easy to follow using it. So now I'm going to just give you an example of how to make this kind of figure. I don't know about the time. Should we take a break now? Maybe I will take a break, and I will come back and we make this exercise, and then I think in 1 hour we can wrap up everything. So let's have a small break, please. Any question? No. So welcome back. to the exercise how to make a figure. So the layout is going to be to open your vectorial drawing software, create a new document, insert the pictures, insert the scale bar, calibrate the intensity, save, and export for publication. So I'm going to share my screen, my full desktop right there. And we're going to do that. So I just open up Adobe Illustrator, and now I'm going to create a new document. So there is things that are already saved, but I always ask you to kind of look at the settings. So the size is okay. You see it's a letter size. You can change the orientation of the image. This is the bleeding. You can include it, but I usually do it myself. So I don't. This is just a space between the side of the image, side of the figure, and the working space. I usually do it myself. Here you have to select the RGB color and raster effects to be at least 300. And then you can create. So now you have your figure starting here, and you can zoom in and out, and you can actually import your images. In Adobe Illustrator, everything works with layers. So you have different layers on top of each other. You can actually add one or two layers as you need them. I'm gonna just import the image I made before. So if you remember, I made this image called cropped RGB. So I'm gonna take it and I'm just gonna drop it in my image. As you can see, the image size is much bigger than actually my file, because we have a very high resolution here. You can also see that there is a cross on it. This cross means that it's actually a link. This picture is a link to the file. What I want to do is to actually embed the picture inside. So I'm going to click embed. So now if I save my file, the picture will be inside the raw image picture, and there is no compression there. So I'm going to save as Figure 1, Illustrator. There. Here you can check different options. I always propose people to create a PDF compatible and to include the ICC profiles. This compression is not with loss. It's actually compressing the size of the file, but without losing data. So you can safely leave it on. I'm going to press OK. So now I have this big figure, but it's much bigger than what I can use. So I need to resize this image. And I'm going to resize it to maybe 111 points. Oops. If I do that, you see that the ratio, the image has been modified. So this, you don't want to do that. So you have to lock the ratio between width and height. And you can do that by clicking this button here. So now I resize, and now it has a nice size. That's my image. I can move it around, and maybe I'm gonna put it on the top left here. There. Then you want to add a letter. So I'm gonna add a letter. So I click text. I click on the image, and I'm gonna put A. And you see that by default on the Illustrator, the font is Myriad Pro, but in my case, I like Calibri. Obviously, it's not there, so I'm going to use Arial. There. So I have my Arial image. You might actually want to put it in white and put it above the text, like that. Good. The second thing I want to do is maybe import another image. So you can do exactly the same thing. Maybe I'm going to add this one. It's a completely different image. Doesn't matter in this case. I'm going to embed it. I'm going to put it on the side there. And now maybe I'm gonna add a B. To add a B, you can just take the letter you have and move it while pressing the Option key, and it's gonna duplicate it. You can see that it's behind this image. So if you want to bring it to the top, you have to send this image to the back. So you click on it and you get just sent to the back. How do I do that? Reset essentials. Okay. Actually, I don't know how to make it to the back. Où ça?
T'étais au bon endroit, c'est arrange, je pense.
Ah, ici, c'est ça. Merci, merci. Je sais plus qui parle, mais merci. And then you can put B. There you go. Okay, the next thing you want to do is maybe to add the scale bar on this image. So the scale bar, there is two ways of doing it. Is either your image is calibrated, so you know how many pixels, how many micrometers per pixel. Either your image is not calibrated. If it's not calibrated, then you have to take a different image with a micrometer slide, and I'm going to show you what it looks like. So my images are not calibrated, so I went to the microscope using this little slide, and I took a picture of that. You can see that this calibration bar is not really well made. It's a bit bent. So something very important is actually rotate this so it's perfectly horizontal. So I'm gonna go Image, Transform, Rotate, and I'm gonna put a preview, and I'm gonna do maybe 1.5. Maybe this too much. Yeah, this looks good. 1.2. So yeah, you need to make sure that this scale bar or this micrometer slide is taken really horizontally. Otherwise, it's gonna mess up your calibration. Not too much, but it's gonna still mess up your calibration. And I'm gonna save as a TIFF. Good. Now, if you remember, I cropped my image. So this image has been cropped, the original image. And you can see how much has been cropped. It's 2094 pixels. So what I should do is actually crop my image in the same way by making it 2094 square. Sometimes it's hard to make a square that is this size. So you can actually use the macro recorder to make it. So if you use the macro recorder and you make a square, it's going to tell you your square is, your square is that size. You see, it tells you in pixels, your square is that size. So what I want is to create a rectangle that starts at 0, 0, and the size is 2094 something, 2094. 2594. There. So if I run this macro now, it's going to make exactly the square I want to do here. And I'm going to move that something here and crop and save. So then you can import this image temporarily, the crop images here. You can embed if you want. You can resize it to the same size, which is 111. And now I know what this image corresponds to in the real life. So what I want is to add a little square or a little line, but it's going to measure exactly the length here. So this is 100 microns, and this is 200 microns. So maybe I'm going to take 200 microns, like that. I'm going to put the color in. So inside is going to be white, outside is going to be nothing. And I'm going to put it there. When you say, oh, it's a lot of work for an image that is it's calibrated, yes, it is. But usually you have all your images the same way, so you can just do one, and then all the others going to be the same. Oops. So I'm going to write the text here. And the text is 200 microns. I'm going to put it on the top and maybe change the size of the font. Maybe go to 10. There we go. Good. When you say, okay, if I want to arrange everything, if I want to change the size of this thing, I will have to change the size again of my scale bar. Not really. You can actually select everything, group them, and if you resize it, it will actually also resize the scale bar. So you don't need to worry about that. So that's done. I'll put it back to the original size. The important thing about vectorial drawing is that this bar will always look super sharp. So if I zoom in, the maximum I can zoom in, it will always look sharp, because the way it's made is not a pixel. It's a mathematical formula that tells draw a square here. So the same thing with the text. First of all, it doesn't take space, so the size of the file will be limited because of the things you added on top. And the second, you will never lose information. So what's taking space in the figure are the actual raw data images. The second thing I want to show you is how to add maybe an arrow or something like that on your image. And you can just take the segment tool, or line segment, and just draw an image, a line. Then you have to tell, okay, this line is going to be white, for example. And at the end of the line, I want to put an arrow. And how do I do that? I don't remember now. Oops. Should be in window. Oops. I can't remember how to make it, but you just have to define the end of the line. I think it's appearance, maybe. Yeah, here. So you select appearance, stroke, and then you're going to say at the end, I want this kind of line. You can also say, I want the line or the arrow to be a bit smaller than the actual size. And then you can take it here and move it there. Good. Then some people is going to ask me, oh yeah, sometimes you do that, but then you have to split the channel. You have to show the same thing on the current image. So let's do that for a sec. To split the channel, you don't use your RGB image, you use the original that is cropped, and we're going to do that. So I open it with image J. I'm going to do edit, image color, split channel. So I have my 3 channels here. I'm going to display them into a grayscale. So I'm going to change the LUT, lookup tables, gray. And again, I'm going to save the image itself. So this is C1. This is C2. Lookup table, gray. And I'm going to save C2. There is a microphone that is on. I think it might be cool to turn it down. And Isley, do you want to turn it on? Thank you. So I have my 3 images now that have been saved. I'm gonna bring them inside the Illustrator. And they are here, C1, C2, and C3, and you can bring them here. So of course you have to embed all of them, and they are much bigger than what they should be. So I'm gonna put 111, the same size than the others. And then I'm gonna move back there. So one other thing you want to do is maybe put the arrow exactly at the same place. And there is two ways of doing that. So I'm going to put all of them here. This I can remove, and this I can remove. The first way of doing it would be to actually put this guy on the top of each other. This is easily done, and there is how do we say? There is a snap tool. So Illustrator brings you the thing exactly on the same place. Then you can actually decide not to display the image itself, but to display only the kind of the skeleton. And if you do that, then you can select the thing that is behind. So I selected both of them. I'm gonna copy them. And if I send now this guy to the back, I should have my arrow exactly the same place. And that's the first way to do it. The second way to do it is to use the rulers and the guides. So the rulers are the things we can display on the side using the command R. And you can put a guide just at the edge of your image. And you can also put a guide at the tip of your arrow. Then you can take these guides and move them together on the side of your image. And then you can take this arrow and bring it and copy it and bring it exactly at the same place. So I'm going to do that for the third image.
Nicolas?
Ouais.
On a un commentaire dans le chat, je ne sais pas si tu l'avais vu.
Non.
Il y a quelqu'un qui a dit, François Auclair dit, ça ne serait pas plus facile pour un éventuel lecteur de mettre les channels dans les mêmes couleurs que le composite?
Alors, oui et non, c'est un choix personnel. Si tu les mets dans les mêmes couleurs, on voit moins bien les bleus que le reste des couleurs. Donc, ça va peut-être être un peu... on va pouvoir apprécier les couleurs différemment. Donc, c'est vraiment un choix personnel. Ça se peut très bien. Moi, j'ai choisi de ne pas le faire dans ce cas précis, mais ça se peut. Fait que j'ai mis la deuxième image. I put the arrow on the second image exactly at the same place using these guides. And because the image are aligned horizontally, you can also use another guide if you need to. I know that the arrows are aligned horizontally. So that's how you do that. The same thing can apply for shapes that are not, I would say, just a line. If you want to make maybe I'm going to take back the spinal cord that I was using before. Where is this table? Adjusted C2, C1. Well, I'm going to take the C1. Okay, I'm going to embed it. So I have this spinal cord, and maybe what I want is just to draw a layout around it. This is a bit more complex than just making a line, but the concept is the same. So you have to use this tool. It's called curvature tool, and you will go to your image on the top of your image, and you're going to click. And when you click, oops, it's not the right tool. It's a pen tool, actually. You're going to click. And when you click, you see that you can actually hold it and extend it. And what this creates is not a point. It's a point plus a vector. A vector is kind of give you the force of where is the line going. And if you release it and then add another one, you see that the vector is now tangent to the line I want to draw. So if I make another one, and another one, and another one, I can draw like that lines that are curved. So this line is just the skeleton. On the top of it, I can add, for example, black. I can change the color. I can also change the stroke. If I want to be dashed, I can choose how the dash is made. Maybe I want to have you know, a big space between the dash. I can also say I want it round up. You can also put an arrow if you like to, and you can also say, actually, I don't want it to be flat. I want it to be with a kind of a thin shape at the beginning and a heavy shape at the middle. So this you can all change. But what is important is it's just the skeleton. So I'm going to make one because usually what you want to do as a skeleton is the contour of your image, and you can do it making these lines like that. But to look kind of professional, don't make too much vectors, and we can use a function at the end to kind of make it cleaner. I will show you this in a sec. So I'm drawing this. And I'm closing it. So what I can select here, it's actually to evenly space the dash. Otherwise, I'm gonna have these two guys next to each other. So maybe I don't want that. I'm gonna choose the white. And also what I want is maybe simplify the things I've drawn. If you draw a little area that is very complex and it's not like very, how do you explain that? But it's not very accurate. You can actually choose to simplify it. And I'm showing you in a sec how to do that. So here you have the layout, you have the skeleton with the points on it. You can go to object, path, and you can decide to add anchor points like that. But you can also add, ask to simplify your shape. And when you simplify it, you can actually choose how much simplify you want it to be. So you can actually simplify to the minimum, and it will change the shape of what you are looking at. It can also show you how much is going to change. You see that here the line is going to change a tiny bit. So by using this, you can have a kind of a more professional looking image. One other thing you can change also, it's and I want to show you that because you can spend a lot of time searching for that. So you see that this anchor here was kind of like that. So it was making a shape that is not very clear. So you can actually choose and kind of modify it with a kind of a larger shape. Also here you have a very sharp turn. This sharp turn make the display not very nice. So you can actually make it more flat. Something like that. And then it is going to look nicer on this side. So if you compare this side to this side, you see it's nicer. And then you will place it on the other image exactly the same way you have done for the first for the arrow. So if I save my file, it's going to take a bit of time to save the file because now there is like 4 images inside. When you want to send it to your boss, you don't want to send the full resolution image, so you're going to export the image. You can also use save a copy. Both of them work. I'm going to show you maybe save a copy first. So if you save a copy, you can choose a PDF, which is a very nice format because image inside is going to be compressed, but arts like the arrows will be kept under the vectorial format, which doesn't take space at all. So it doesn't matter how much you're going to zoom in, the lines and the text is going to look very, very nice. Only the image will be compressed. So when you click the save, you have this option. And usually people just press save and just don't care. But in this case, since I want to send it to my boss, I'm going to just remove the editing capabilities, optimize for fast viewing, and also I'm going to compress the image. And here you can compress the color image and also the grayscale image and the monochrome image. So I'm going to actually just select all of them. And I'm going to select bicubic downsampling. Bicubic is more takes more power to calculate the downsampling, but it's with the regular computer, it's kind of fine. Then you can decide how much PPI the image will be brought to. And in this case, you see that it brings everybody to 150 PPI. And I'm going to click save. And I should have on my desktop the image that is saved. So I have my two files here, figure 1 and figure 1 Illustrator. I can open it with a regular software, like Preview for Mac, and I can zoom in. And if I zoom in, you see that the quality of the lines doesn't move. It's always super sharp. Whereas the quality of the image has been downsampled. So this is kind of a less resolution. So when you're gonna send the data for publication, they're gonna ask you a specific file format. It can be EPS, it can be SVG, it can be PDF. At this time, when you want to send the high resolution, you will choose Save As, but using a 600 ppi resolution for the color and the black and white images. The file is going to be bigger, but that's what you want for the presentation. So just to give you an idea of the size, this file is 21 megabytes, and this PDF is downsampled, so it's only 180 kilobytes. So it's very small. So this is totally sendable by email without losing the resolution for the arts that you've put on the top. Okay, I think I'm gonna stop here for the demo. Any question on that? No, then I move to the next part, which is writing materials and methods for microscopy or in microscopy. And when I was writing it, I just realized that there is a lot of useful information that should be included in the materials and methods. In short, it should be included everything that you have used for your experience. Everything that is involved in your experience should be there. And this includes the biological, the fluorophores, how you prepare your samples, which mounting media you're using, everything about the instrument, but also how you used the instrument, and of course, how you treated the images, or how you processed your images. And I was writing it, and then I think it's super long and super complex. And so I kind of stopped doing the list, because I think for you guys, it's going to be a pain. So I started making the list, and then I stopped. And then I say, okay, what are your options to write materials and methods? How you can actually do that? Well, there is 3 ways of making it. Instead of making a list myself and trying to present it one by one, I just say, okay, well, how you can make it. So the first one is to actually fill up a template that has been made by someone else, and you can just, fill in your data. The second one is a very nice tool, and I'm going to present it in more details. And the last option you have is to actually ask people, if you have a microscopy platform, come and ask them to revise and read, or ask them for help to write it, because they know and they have done it a number of times, so they exactly know what to put in. This have been taken from Harvard Medical School, and they have an Nikon imaging center. And the Nikon gives a template, and this is the template they give. So you can actually copy and paste this, and you can fill in anything that is between quotes or brackets. So here in the middle, you see that you have insert confocal or TIFF model of microscope, because on this platform, it's all Nikon TIE inverted microscope or ATI. This is already there. But you can also insert the objective lens and also the fluorophore. Also, they state that you should put the exact variant. So do not put GFP, because there is different kind of GFP. There is MGFP, EGFP, and so on and so on. So they give you a nice template to fill it in. So that's the first way. The second way is to actually use this tool that has been published recently, and it's been done by Rebecca Senft. It's called MicCheck, and it's been published in this article, Best Practices and Tools for Reporting Reproducible Fluorescent Microscopy Methods, and I give the link here. We're going to click on MicCheck, and I'm going to show you what it looks like. So you copy the link, and you go to MicCheck here on your favorite web browser. And you obtain this page. And here on the left side, you can actually check what you are been using. So in this case, my microscope was a wide field microscope. I was using for emission filter cubes. It was not a laser light source. It was a excite lamp. I didn't use bright field. I didn't use additional magnification. It was a multicolor image, and I didn't do multi-point. I didn't do anything of that. It was not live imaging. I was looking at fluorescent organic dyes, and I didn't express anything. It was no, actually, yeah, I didn't express anything in my my sample. And yes, I was part of the core facility. And when you have selected that, you see that you have a checklist that has been built, and you can actually click to show example of what she meant by each of these conditions, and you can export this as a PDF. So now you have a checklist to write your metadata and write to your materials and methods about your microscope. So you can start writing saying, I was acquiring the images using a microscope from Leica, or in this case was an Icon. The illumination shutter was from Uniblitz. The manufacturer of the stage was Nikon, and it gives you everything you should actually fill in. So it's quite broad, but it's a very nice tool because you won't forget anything important. And when I was preparing this presentation, I sent her an email, and she told me that actually she's making a new MIC check version 2, I guess. And she sent me the link for the preview. So I guess, you know, there is only a few people that have seen this except Rebecca and me. So we're going to give it a try and see what's different in the new app. Yeah. So she takes the same template where you have to, you know, to answer the question about the thing, but there is also this select a microscope core and select a microscope here. And then these things are actually linked to a database that has not been made by Rebecca, but made by another guy. So I'm going to give you the example of what she means by linking this. By linking this, you can actually check directly which filter you have been using. So it's actually writing your materials and methods itself, and it brings you the data you are interested in. So that's the example of how things get better. So our tool was nice, but you still have to write everything on your own. Now it's kind of including the data directly from the microscope itself. But this data has to be made, and this comes from another article published also in Nature Methods. It's called Micrometa. It's not meant for you guys, it's meant for people in the core facility to build some kind of file that will be usable by that. And also you can actually report them directly. Okay. So where's Micrometa? It worked yesterday, so I'm gonna just reopen it. Yeah. Okay, so in Micrometa, what you have, it's two options. You can manage an instrument, or you can manage settings for an image. If you go to manage instrument, you can load it from a file. And in this case, my file, it should be in my OneDrive, desktop, Atelier Microscopie, Micrometa, and the microscope is this guy. Continue. So Micrometa, what it does, it just builds a microscope and all the parts that belongs to this microscope, and you can actually specify them, each of them individually. So this is a triple pass dichroic for green, red, and far red, and you have the manufacturer specification, the field, and everything is including that. So it describes really the microscope, and everything is there, including the softwares and the modules and everything. So this obviously will take a lot of time for a regular user to do it, but for a platform or for core facility, you can generate this file, and then you can make it available for users like you guys So they can actually take everything from the microscope and they don't need to think about it because they can just export this microscope into a file, and you put your figure 1 microscope, and this JSON file is generated, and everything about the microscope is inside, and you don't care, you don't write anything in the materials and method, you just publish the JSON file. So obviously this has to be made by your core facility, but where it gets nicer is that you can put this JSON file inside this this checklist generator, MicCheck, and you can actually write your materials and method directly. So this is in current development, it's not there yet, but I thought it was a very interesting tool to write the materials and methods. Okay. So I talked about Rebecca and give the demo for Micrometa and for MicCheck and MicCheck 2 and Micrometa. And the last way to actually write your materials and methods is to ask the experts. There is microscopy experts and in different locations. So Monique Vasseur, that is with me. There is also Chantal Delille. There is people at the CHUM, at HMR, ICM, RCM, ERIC, and Saint Justine. So all of these people are experts and they have contributed to a number of articles. So you can contact them. They are meant for that, to help you write your materials and methods in a good way. Because to come back to the beginning, you are responsible for the data you publish. So don't hesitate to get help from the people they know the best or they know the most, and they will help you to write the good materials and methods for that. That's it for today. You see that it was much shorter than the previous presentation, and I was trying to cover a bit of everything, how to deal with ethical aspects of microscopy, what obligation you have to follow, how to create a figure, prepare your image and compose the figure, and also what to and how to write materials and methods. So yeah, that's the end for today. And if you have any questions, you can always reach us at microscopie-facmed@umontreal.ca. It's either me or Monique or someone else or Chantal will reply to you, and we will be very happy to help. And that's how the first workshop for microscopy ends at the UDM. Thank you.